So friends, we have discussed the mechanism of electroporation, that is how the passage of electric current causes the formation of pores in the cell membrane, and what are the various factors that can affect the efficiency of that electroporation process. But how we determine that efficiency is by quantification of the electroporation, and there are different methods of quantification this electroporation process, and these are the electrical conductivity, by measuring the electrical conductivity. And it has been found that the electrical conductivity increases as the cells are damaged. So we can say that the electrical conductivity will be directly proportional to the damage of the cell caused by the electric pulses or the current pulses. So higher the electrical conductivity of the media, higher will be the damage. Second technique is the measurement of the diffusion coefficient, and again with the damage of cell, the diffusion coefficient also increases, because as the cell membranes are damaged, the diffusion of the components into the solvent increases, thereby increasing the overall diffusion coefficient. But it also depends, this diffusion coefficient, one has to be very careful, because diffusion coefficient and also electrical conductivity, these are dependent upon the moisture that is present in the cell or in the medium where this process is being taken. So one has to be very careful while measuring electrical conductivity and the diffusion coefficient. Second, the diffusion coefficient method is also dependent upon the condition of the sample, whether the sample which is being used for the extraction is intact or it has been thawed or powdered, because in a study it was found that when a freeze-dried thawed sample of potato was used for extraction using pulse electric field, there was a significant increase in the diffusion coefficient in comparison to the sample of the potato which was simply dried and then used. So later it was hypothesized that freeze drying and thawing of the potato sample also causes the damage of the cell membranes, thereby increasing the overall diffusion coefficient, thereby increasing the overall extraction yield. Therefore, such kind of parameters, they largely also depend upon the condition of the sample, whether it is intact or whether it is already damaged. Then the third method by which you can quantify the electroporation is by studying the textural characters of the tissue that has been exposed to the pulsed electric currents. It has been observed that whenever the tissues or the cells are exposed to the pulsed currents or the electric field, it causes the loss in their textural strength, their elasticity, thereby increasing the overall damage to the cell. So if there is a significant decrease in the textural strength, there is a significant decrease in the elasticity of the cell, that indicates significant damage has been done by the electric current onto the cells. Another method is by observing the cells or the tissue under microscope. So in this figure you can see three different rows. The uppermost or the topmost row is of the sample that has been exposed to the pulse electric current. The middle row is of the fresh sample, and the third row is of the dried sample. And you can see each row has three different images. The first image is at 2000 times magnification, second image is at 10,000 times magnification, and the third one is at 50,000 times magnification. So you can clearly see in images of the sample that have been exposed to pulse electric field, in the third image, that is 50,000 times magnified, you can clearly see the disruption of the surface of the cell. You can also clearly see the formation of small pores on the surface of the cell, which clearly indicate that the cell has been damaged and the cellular contents have been extracted. Whereas in the fresh sample you can see the structure completely intact, and you can clearly see the surface characters of that particular structure also. whereas in case of dried sample you can see slight change in the surface characters of the sample, but yes, the tissues or the cell are almost intact, but slight change in their morphology. So this is another way by which you can clearly measure, clearly see whether the cell has been completely damaged by the current or not. Now we move on to the second mechanism by which the electric current causes damage to the membrane or causes the opening of the cells, and this is Joule's overheating. And as I told you that whenever the current is passed through a conductive media, there is a resistance towards the flow of that current, and this resistance leads to the generation of Joule's heat, which eventually causes the warming up of the media. And this increase in temperature of the media is directly proportional to the time and the intensity of the current that is passed through that media. And if the current is passed for a longer duration, then obviously it will cause overheating of the membranes, which eventually lead to their damage. The third mechanism is the electro-osmotic transportation. Now, electroosmosis, as the name indicates, this is a phenomena where osmosis occurs with the help of electric current. And in this phenomena, there is a direct electrolyte flow near the charged surface in a pore under the influence of external electric field. And the velocity of the electroosmosis, again, it will be directly proportional to the strength of the electric field. Now this causes a slight change in the balance of the chemicals that are present in the cell, because now these chemicals or the ions, they are moving under the influence of electric current, and this shifting of the chemical balance or production or induction of the chemical imbalance due to the electrolyte outflow causes the lysis of the cells, and all the cellular components then they get solubilized in the solvent that is present in the media. So this was the third mechanism by which the current pulses causes damage to the membrane. Now we move on to the instrumentation of pulse electric field technology, and there are different components that are involved in carrying out this technique or in manufacturing or designing the equipment for this technique. First one is pulse generator, which is considered as the heart of the pulse electrified instrument, because it is responsible for the generation of the pulses, electric current pulses. Second is the extraction chamber. As the name indicates, here the plant material or the sample from which secondary metabolites have to be extracted is kept. Third is the thermostat, which is required to maintain the specified temperature. And fourth is the pump that is used to pump the compressed air into the extraction chamber, thereby causing the pressing of the sample, which further enhances the extraction efficiency. Apart from these four major components, various types of sensors and valves are involved in designing of this instrument, so that if there is any increase in the current intensity beyond the specified limits, if there is any increase in the temperature beyond the specified limits, the whole process can be checked. So coming first on to the pulse generator, as I told you that it is the heart of the pulse electric field extractor, and it provides or it produces the electric pulses. Now these pulses which are produced by this generator are of desired voltage, they can be of desired shape, and they are produced for the specified duration. And all these steps, they are controlled by different components that are present in the generator, and these different components come together to form a complex structure or complex network, which we call it as pulse forming network. And the different components under this network are, it is composed basically of one or more DC power supplies which help in inducing the electric currents, a charging resistor which basically helps in controlling the intensity of the electric current, Then a capacitor bank is there, wherein two or more capacitors are connected in parallel, which are connected to different switches in order to store the current in them. And lastly, this pulse field network also contains a pulse shaping inductor and resistor. Now this pulse shaping inductor and resistors, they help in providing the pulses of desired shape, because there are different types of pulse shapes that can be used in this extraction techniques, and depending upon the type of the shape of the pulse is required, different generators are there. Now depending upon the maximum output from these generators can be classified into the benchtop pulse generator, which can provide a maximum of 2.5 kilovolt of pulses, and the second class is the lab scale pulse generators, which can provide up to 40 kilowatts of the pulses. And in lab scale pulse generators, capacitors are used because we are using a high intensity pulses, so capacitors are used here in order to store that particular current. Now, as I told you that the second portion is the extraction chamber or the treatment chamber. Now, on the basis of the mode of extraction, now we have discussed different extraction techniques, wherein we discuss about the static mode and the dynamic mode of the extraction. So same two types of modes also occur in pulse electric field technique, and depending upon the technique or the mode you are using, we have different chambers. So we have batch systems which are used for static mode, and in batch systems we have to load the sample into the extraction chamber, expose it to the electric current at a specified intensity for specified period of time. Once the material is exhausted, the process is stopped, the extraction chamber is emptied, and it is again reloaded with the fresh sample. And since we can extract out a specified amount of the material in this system, these are known as batch systems or the static mode extractors for the pulse electric field technique. These batch systems have a major limitation of the use in the industry, because every time one batch is exhausted, you have to stop the whole process, again recharge the extraction chamber by the fresh material. So this stopping and recharging, this is not very acceptable at the industrial scale, because industries always go for the complete automation of the process, wherein they don't have to stop the process to recharge a particular extraction cell, or they have to stop the process at a particular step. For such scenarios, we have continuous systems which work on the dynamic mode, wherein we optimize the flow of the material through the extraction chamber, so that you don't have to stop the entire process in order to feed the extraction chamber with the fresh material. So the flow of the material, plant material, is optimized in such a manner that the process continuously goes on, and therefore these are known as continuous system, and these are more popular at the commercial or industrial level. Apart from the basis of the mode of classification, we can also classify the treatment chambers or extraction chambers on the basis of their design. And on this basis, we have different types of extraction chambers, like parallel plate chambers. These are largely used for static mode. We have coaxial chambers, which are largely for continuous mode, and we also have collinear extraction chambers, which are again used for the continuous extraction modes. Now in this image, you can clearly see different extraction chambers on the basis of the arrangements of the electrodes. So on the image on the left side, you will see the first and the topmost image is of the parallel plate chamber, wherein the two electrodes are placed parallel to each other. The second, or the B portion in the image, is the simple continuous chamber, wherein the flow of the material is optimized. It enters from one side and it exits from the opposite side, and the process continuously goes on. Third part of this image is the coaxial chamber, and fourth is the collinear chamber. On the image on the right, you can clearly see the arrangement of the two electrodes in the parallel plate chamber, and you can clearly see the arrangement of the electrodes in the coaxial chamber, wherein one electrode is at the center and the second electrode surrounds the central electrode. So this forms the coaxial chamber. Now this is the overall outlay of a pulse electric field extractor, wherein we have a data processing unit by which you give the commands, you set up the intensity of the electric current pulses, you set up the duration for which you are exposing the material to those current pulses. Then this command is given to pulse generator, which generates the electric current pulses of that specified intensity for specified interval of time. These pulses are then shifted onto the extraction chamber, and you can see the two electrodes are present in the parallel position to each other. Therefore this is a parallel plate extraction chamber, and you can see the plant material is placed in the extraction chamber, current is passed, the cells are lysed, the cellular contents are extracted, and the contents get solubilized, or the juice is extracted, and you can collect that juice at the bottom. Now the bottom plate of the extractor is replaced by a perforated stainless steel plate, which also causes the filtration of the juice. So most of the vegetative debris it left behind in the extraction chamber, and you get the filtered juice. Now another improvement in this setup you will see is the application of the pressure onto the sample that is placed in the extraction chamber, and this pressure is applied with the help of compressed air. Now it has been observed that whenever pressing of the plant material is done along with the pulse electric field or the current pulses, the extraction efficiency improves, and the yield of the extract or the juice, whatever we are extracting, it improves tremendously in comparison when this technique is used without pressing. So this is a picture of a pilot scale up of pulse electric field extraction, wherein spinach was used to extract out its constituent. You can see the pulse generator is there, filter press is there, which filters the material, which presses the material once the extraction, once the current damages the cells or damages the membranes of the cell. And the images on the right, you can see the pressed cake which is obtained after the extraction is complete. Now we move on to the various factors that affect the extraction efficiency. Now these are the electric field intensity, shape of the pulse, because we know that there are different pulse generators depending upon the shape of the pulse that is required. The efficiency also is affected by the time duration for which pulses are being passed through that particular media. Then pulse interval, that is the time duration between the two pulses, also affects the extraction efficiency, because if there is a significant increase in the time duration between the two pulses, then we are allowing the cells to reseal themselves, because we know that there is a natural tendency of the membranes to reorient themselves once they are slightly damaged. So if we increase that duration between the two pulses, we are giving sufficient time to the cell membranes to reorient themselves, which causes closure of the pores that are formed in the membrane. So one has to be very critical. We will discuss this in detail also. Then the next factor is the type of the solvent, concentration of the solvent, and the pH. We know that secondary metabolites from the plants which we want to extract, they have a varied chemistry, they have a varied chemical structures. So depending upon their chemistry, depending upon their chemical structures, different solvents and more specifically different pH also has an impact on the extraction efficiency. Next factor is the solvent to feed ratio, that is how much solvent we are using to extract out a particular amount of the plant drug material. If we increase the solvent to feed ratio, obviously we are increasing the volume of the solvent that is required to extract a given amount of the sample. Again, here the economical aspects come into play, because if we increase the volume of the solvent beyond a specified limit, then the process becomes uneconomical, because a lot of energy will be required in order to concentrate, in order to evaporate that solvent. So one has to be very critical in optimizing to solvent to the feed ratio. And last but not the least factor is the temperature. And we know that the increase in temperature basically helps in solubilization of the components, helps in increasing the mass transfer rate, thereby overall increasing the extraction efficiency. But again, one has to be very careful while adjusting the temperature, because certain constituents are liable to thermal degradation. So we don't want to degrade our constituent just in order to increase the extraction efficiency. So one has to be very careful while optimizing the temperature conditions also. Now we will discuss these factors in detail. So coming on to the electric field intensity, and as I told you that as we increase the intensity of the electric current, there is more poration, more polarization of the membrane. More polarization means more pore formation, and more pore formation in the membrane means better extraction yield or the extraction process. One has to be always keep the basic thing in mind, that the intensity of the electric field or the current that has been selected should be above the threshold value but below the critical value. And we discussed that the threshold value of 0.5 volt to 1.5 volt. So the intensity should always be higher than 0.5 volt, because below that intensity the current would not be able to form the pore in the membrane of that particular cell. But the intensity should be below the critical value, because if you increase the intensity above that critical value, then the pores that are formed, they cannot be reversed. The membrane would not be able to reorient itself if they are exposed to the intensity above the critical value, and this critical value varies from the type of the cell or from cell to cell. Now the intensity of the electric current, it also depends upon the width of the pulse, and width of the pulse means it is the time taken by one complete pulse to pass through. And it has been seen that the intensity of the electric field decreases with the increase in the width of the pulse. Say for example, if the width of the pulse increases to 50 milliseconds, then the intensity of the electric field decreases to 4.9 kilovolt per second. Whereas if we decrease the width of the same pulse to 2 milliseconds, then the electric intensity increases to 40 kilovolt per centimeter. So this is how the width of the pulse impacts the intensity of the electric current. So bottom line is, one has to be very careful in optimizing the width of the pulse. More the width of the pulse, lesser will be the intensity, and lesser will be the intensity, more time duration will be required, or the poration will not be proper. Whereas if the width of the pulse is too small, the intensity of the electric field will be too high, which can lead to the irreversible damage to the cell membrane. So we have to optimize this. Then extraction yield, it has been seen that it increases up to a specific level of the electric field intensity, and then gradually it decreases. And as we discussed that whenever the current is passed through a conductive media, there is a slight resistance in the flow of the current, and this resistance basically causes increase in temperature. And perhaps this increase in temperature was considered as the major reason for decrease in the yield by increasing the field intensity beyond the specified limits. There was a study carried out by Yin and his group in 2006, wherein they studied the effect of increase in the electric field intensity on the yield of polysaccharides which they were extracting out from a frog. So they observed that when the intensity was in the range of 50 to 20 kilovolt, there was around 17 to 27% of the extraction yield of the polysaccharides. But when the electric field intensity was increased from 20 to 40 kilovolts, there was decrease in the yield of polysaccharides from roughly 27% to 21%. and again they hypothesized that this degradation was largely due to the thermal decomposition of the polysaccharides. Therefore, we can say that the electric field intensity should be optimum, and this should be optimum in order to avoid the thermal degradation of the constituents. It should be optimum in order to avoid more power consumption, because if you want to increase the intensity you have to supply more power. More power means more economic aspects come into play, and your process may become economically unviable. Further, the intensity should also be optimized in order to maintain a stable system, because we discussed that there are various sensors that are placed in order to check the increase in intensity, increase in temperature. So in order to maintain the stability of the system or the extractor, the intensity should always be optimized. And last but not the least, in order to prevent the irreversible damage to the sample which is being extracted. So friends, this was about the effect of the electric field intensity on overall extraction process. In the next lecture, I will be discussing the other factors that can affect the overall extraction efficiency. Thank you.